is separating those cells. We're going to go through cytokinesis here. What we ended up with are 46 chromosomes in each of these new nuclei. Those cells that result from mitosis are genetically identical. Over here in meiosis, however, we now have 23 chromosomes that are in each nucleus, but there's a big difference. These 23 chromosomes are still double-stranded. What we're doing here during meiosis is making 23 chromosomes in each cell that are actually single-stranded. We call that process making haploid cells. What does the word haploid mean? Half. Yeah, half the chromosome number, right? What did you make over here in the mitosis process?
Diploid.
Diploid, which is double the chromosomes. That's 46 in each cell. So we're going for, in meiosis, the process making 23 single-stranded chromosomes. So you see that we're not quite there yet. We still have 23 double-stranded chromosomes. Well, with mitosis, you're done, okay? But let's take a look. Meiosis needs to continue because we need to separate those 23 double-stranded chromosomes now into 23 single-stranded chromosomes. All right, go back through prophase. Okay, let's get you into metaphase. Now you have these X-shaped chromosomes lined up across the metaphase plate. There are 23 double-stranded chromosomes across this metaphase plate. There are 23 double-stranded chromosomes up here. Normally, if this was mitosis, there would have been 46 double-stranded chromosomes across the metaphase plate. So you see, we're working our way to getting half the chromosome numbers in each of these cells. Okay. Now this should look familiar. You've now separated those double-stranded chromosomes into single-stranded chromosomes. What we're doing now is pulling 23 single-stranded chromosomes to this end of the cell, pulling 23 single-stranded chromosomes to the top end. We're not just doing it for this cell, we're also doing it for the top cell as well. Okay, and once telophase and cytokinesis are complete, take a look. We've got 4 cells resulting. They are haploid, meaning that they contain a single set of chromosomes. And each cell here has 23 single-stranded chromosomes. The important factor here is when fertilization takes place, you have an egg with 23 single-stranded chromosomes that joins with a sperm with 23 single-stranded chromosomes. What do they get together and make?
46.
Yeah, 46 double-stranded chromosomes. Hey, that's what you made through mitosis. So at that point, you can go back to a full-bodied human being that has the normal number of chromosomes. So this process of meiosis here is called reduction division. Our goal is to reduce the number of chromosomes by half. That way, when those cells unite through fertilization, we're bringing half the DNA from mom with half the DNA from dad together. This process here, if we were to spend more time on it, there's actually the arms of the chromosomes touch, they break off and switch. It's called crossing over. And that's the way you actually get genetic, differences among people that are, close relatives like brothers and sisters. So you get that switching over of genes in this portion of meiosis here. Now, let's take that process and apply it to page 144 and walk through spermatogenesis. Let's take a look. We're going to start with the picture on the bottom of the page, and I'm going to start you with the spermatogonium, which is a stem cell present in the testes of a male. Keep in mind the goal of spermatogenesis is to make how many sperm? 4. Good. What process do they mostly use? Meiosis, good. The goal is to reduce a diploid cell, represented there by 2 ends. Goal is to break that diploid cell apart into 4 cells, each of which is haploid. keep us our running guide over here. When we talk about diploid, we typically mean 46 chromosomes. When we talk about haploid, we use the letter N to represent haploid. That means just simply 23 chromosomes, but as we find out today, that can be either double-stranded or single-stranded. Well, this stem cell in the male testes is known as the spermatogonium. It's going to undergo mitosis because one of the cells that's resulting from mitosis will go back and join the stem cell line. If the two cells that resulted actually continued on to make sperm, the male would run out of stem cells and would have no more forming cells to actually make sperm. So under the process of mitosis, you'll notice on your worksheet I've just followed one cell. And if you think, well, hey, mitosis normally makes two cells, you're right. One of those two cells rejoins the stem line and goes back and forms as a spermatogonium. So this process here, taking a spermatogonium in the male's testes, dividing it through mitosis, forms for us a primary spermatocyte. You might remember that mitosis makes cells identical to the parent cell. So mitosis is going to make a diploid cell. I'm just going to keep track of our processes here as we go. Now we're going to start the process of meiosis. Meiosis is a two-phase process. What we're going to do now is divide that cell twice in an attempt to reach a haploid state. So you notice that once you go through mitosis, you produce what we call a primary spermatocyte. This primary spermatocyte will undergo meiosis 1, and the naming scheme here is pretty common sense. That produces two secondary spermatocytes. Okay, so we just saw the animation of how meiosis can break those cells or break those chromosomes apart. So as a result of meiosis 1, we now end up with two secondary spermatocytes. Sure, we have reached the haploid stage, but note, these are double-stranded chromosomes. We need for the chromosomes to be single-stranded in order to successfully fertilize an egg that's also haploid and single-stranded chromosomes. So nothing we've made so far is a viable sperm, and the whole bottom line reason is what? Double-stranded, yeah. We have too many chromosomes for any of these cells so far to be a viable sperm. Alright, now we undergo meiosis 2. Meiosis 2 further divides those chromosomes. If we've already reached the haploid state, that's what we want, but there's something wrong with the chromosome stranding. We have double-stranded chromosomes in the